Good afternoon, and thank you for coming to the BSSR lecture series. My name is Mike Spittle, and I'm at OBSSR, and it is my great pleasure to introduce you to Dr. Tom McDade. Briefly, Dr. Tom McDade is a biological anthropologist who conducts research on health and human development in relation to social and cultural contexts and processes. He received a BA in biosocial anthropology at Pomona College, a PhD in anthropology at Emory University, and was a postdoctoral fellow at the Carolina Population Center, UNC Chapel Hill. His work is focused on three topics: impact of social stratification on stress and health, life course perspectives on immune function and the regulation of inflammation, and the integration of biological measures into population-based social science research. It is my great pleasure to be able to introduce you to him, and his talk is called Early Environments, Inflammation, and the Transgenerational Perpetuation of Disparities in Health. Thank you.
Thank you all. Can you hear me? Is this working? Okay. Pleasure to be here. Thanks very much for the invitation. And it's great to see some familiar faces and meet some new ones that I've known over email or over the phone. What I'm going to try to do today is talk about really one of those threads in my work, but they actually come together a bit in this project. And talk about a life course, and what I'm moving towards is an intergenerational perspective on inflammation and social determinants of health. And so much of what we understand about human physiology, biology, development, and health is done with research on laboratory animals, animal models, and in clinical settings in the United States and other more affluent industrialized settings. But as an anthropologist, I'm fundamentally interested in a broader perspective on human variation and how that matters to our understanding of human biology. So the meta message of the talk that I hope you'll take home is that a comparative approach to understanding human biology can actually generate insights into those basic clinical pathophysiological processes that we're interested here in on your campus. And as an anthropologist, it's one of my great privileges to be able to go to some of the places that I'm going to show you about today and to do things in interesting parts of the world. So you're all familiar about this minor revolution in our understanding of health and our increasing emphasis and understanding of the early life origins of health in adulthood. And in fact, it's been almost 100 years that we've known from cohort data coming out of the UK that environments in infancy predict longevity, life expectancy, and disease risk in adulthood. And over the past 20 or so years, David Barker, coming out of England, has been most sort of successful in promoting this as an important area of study. And this has more recently captured public's imagination with a couple of recent high-profile, more public pieces on this issue. And the way the paradigm works, which I'm sure is familiar to most of you, we used to think of health in adulthood as primarily a product of our individual genetic endowments, our current environments, and our lifestyles, and perhaps their interaction. But now we're increasingly coming to appreciate the important roles that environmental exposures early in life have in terms of programming key physiological systems which have direct implications for health in adulthood, and also may moderate the effects of environments and genes on health in adulthood. Now, the vast majority of this research has focused on cardiovascular risk, metabolic diseases, and early sort of nutritional exposures, but we're moving beyond that quite quickly. Now, related to that, we have a long-standing tradition in the social and behavioral sciences in appreciating the impact of early environmental quality, early childhood environments on health, but also on cognitive function, on social function, on emotional function, things that matter to social attainments that we care about. Folks like Jim Heckman, Alberto Poloni, Jack Shunkoff, and others have been aggressively promoting a policy agenda based on this long-standing body of research, emphasizing that investments early in life have greater impact, greater returns than investments later in life. These fields are converging, and it's a very exciting time, I think, to be involved in life course and intergeneral research related to health and its social determinants. And this is an area that I'm moving towards, and if there's time at the end of the talk, I'll talk about two new projects that I'm launching that are related to this. But we can start to think about social environments and inequality or variation in those environments in one generation, how they impact health in that generation, but also how they impact health in the following generation, perhaps through the quality of prenatal environments and early childhood environments, and then health endowments and achievements and developments in one generation can then have impacts on social achievements in that generation and in the following generation. So we're starting to see an articulation of a multi-generational, transgenerational approach to health that I think is going to be quite important as we move forward in this area of population research. Okay. I'm going to move towards my interest in immune function and inflammation, but start by talking about a relatively large body of research on what's called the hygiene hypothesis that's come out of epidemiology and immunology over the past 20 years, which has shown both epidemiologically as well as mechanistically, experimentally, that individuals as adults are at higher risk for allergy, asthma, and autoimmunity, diseases that are related to dysregulation of immune processes, and that individuals who have impoverished nutrition early in life and who have lower levels of microbial exposure in infancy and childhood, mostly proxied by things like whether you have older siblings, whether you lived on a farm, things like that. These kinds of exposures, higher levels of microbial exposure, actually protect you against these kinds of diseases. And the work is really focused on those types of atopic diseases. And the question that I'm going to talk through today is whether this may matter to inflammation and how we regulate to inflammation. 10 years ago, this would have been kind of a niche question, but right now, we are currently obsessed with inflammation, I think both as researchers and in the public eye, because inflammation seems to be the thing that's going to do us all in. It's related to cardiovascular disease, it's related to diabetes, it's related to dementia, to cancer. A number of large epidemiological studies have shown this, and clinically it's starting to be evaluated as well. So inflammation has become this pathological process that we need to control and be concerned about, which is ironic because 15 years ago, our understanding of inflammation was such that if you did not have a functioning inflammatory system, you would die because you need the system in place to protect you against infectious disease and to help healing after an injury. So there's been a change in our conceptualization of inflammation. That's going to feature prominently in the story that I want to tell today. So what has led to this reconceptualization? It's some observations coming out largely of cardiovascular epidemiology, which has shown that if you measure C-reactive protein, a key biomarker of inflammation, acute phase protein, if you measure it and detect it at levels that were previously considered undetectable, but with new assays that are more sensitive, we can measure very small concentrations of CRP. It provides additional predictive risk for cardiovascular disease, in this case heart attack and otherwise healthy adults, than traditional measures, above and beyond traditional measures of cardiovascular risk, like your lipid profile. So here you have your ratio of total cholesterol to HDL. As this goes down, your risk of heart attack goes down at all levels of CRP, but also your level of CRP, independently of your lipid level, higher CRP associated with higher cardiovascular risk, independently, and that's a key feature of this phenomenon. In the population and demographic sciences, inflammation has also gotten attention recently because Eileen Crimmins and Tuck Finch in this paper in PNAS and another in Science have proposed that historical increases in human life expectancy post-epidemiologic transition populations is due to reductions in infectious disease morbidity, which has in turn lowered burdens of inflammation throughout an individual's life course, and what they call has led to the development of a cohort morbidity phenotype. So if you reduce infectious disease in childhood, you reduce the overall lifetime burden of inflammation, and therefore, since inflammation is a contributor to cardiovascular disease, we've lowered cardiovascular risk, therefore increasing life expectancy. So they have some cohort data that suggests this is true, although those data are not without, or their analyses are not without some controversy. It's by no means a done deal, that story. But nonetheless, it's an important part of our current understanding of inflammation. This is what I'm calling the chronic low-grade inflammation model. This is how we understand inflammation in settings like the United States and its relevance to things like cardiovascular disease. And what this is a study of CRP biovariability. And it was an early study that's heavily cited as justification for measuring C-reactive protein one time in an individual to determine their sort of overall chronic level of exposure to inflammation. And these are a set of just 4 individuals who are sampled at 3 or 4 day intervals over a 7-week period, and the dots just connect their values, right? And so what you see, the individuals here at the bottom there's a little bit of noise, but pretty consistently they're low, and certainly lower than these 2 individuals at the top. So if you just do one snapshot in time, you will be able to differentiate these 2 individuals. There's sufficient between individual variation here that this measure should have some prognostic value. So the assumption is that one individual measure of CRP will identify and separate these individuals. And that's an important part of the physiology of the system as well as how we use it epidemiologically. And in terms of physiology, here's how the model works. Adipose tissue, body fat, is a very important source of pro-inflammatory cytokines like IL-6. Those cytokines enter general circulation, and then they go to the liver, cells in the liver, which produce and release C-reactive protein. Chronic infections like herpes virus infections or gingivitis, things like that, may also upregulate these inflammatory pathways and in turn promote the production of CRP. CRP enters general circulation, and then it has these effects that all lead to the initiation and progression of atherosclerosis. And the idea is that these processes are independent of other pathophysiological processes related to high levels of lipids and lipid accumulation and blood pressure damage to the vasculature, things like that. Also, there is some controversy about the degree of causality here, but this is the model that underlies much of this research. Okay, so what do these two have in common? From the look of him around the middle, probably has pretty high levels of CRP. Visceral adipose tissue around the midsection in particular is the most important source of pro-inflammatory cytokines like IL-6. So he probably has elevated CRP, should be increasing his risk for cardiovascular disease as well as other bad things. This is the horseshoe crab. Been on the planet since the days of the dinosaurs. Also has very high concentrations of CRP. And that's because the immune system of the horseshoe crab depends on pattern recognition molecules, aspects of innate immunity as a first-line defense against infections, phagocytosis and things like that. It does not have an elaborated, specific humoral and lymphocyte-driven immune system like mammals do. And in fact, C-reactive protein as a molecule was discovered in 1930 for its ability to bind to the Streptococcus pneumonia bacterium C polysaccharide region. That's why it's called C-reactive protein. So it was first discovered as a molecule that binds to bacteria, to pathogens, and it plays a very important role in all organisms that have it in protecting us against infectious diseases. And from that perspective, this is what the model looks like. It's not a chronic activation, it's an acute phase or acutely responsive model here where you have basically undetectable levels of C-reactive protein. You get challenged with a bacterium, a virus, or an injury that has to be dealt with, and levels of CRP increase by 3 or 4 orders of magnitude within 24 to 48 hours after exposure. That provides a very important, very rapid front line of defense against the infectious agent. And then as the infection starts to resolve and as more specific immune defenses start to come online, which can take 3, 4, 5 days to come online, the concentrations of C-reactive protein drop down to baseline. Okay, so that's an acute phase model. And then, again, in terms of mechanism and physiology, you have a pathogenic challenge. You have pro-inflammatory mediators like IL-6. These mediators induce the liver to produce more CRP. They also cross the blood-brain barrier and induce the symptoms we associate with infection, like fever, loss of appetite, tiredness. And then CRP activates complement, promotes the activity of phagocytic cells, which can clear your body of these infectious agents, and it opsonizes or tags pathogens so that they're more readily recognized by the body as being foreign. And it helps us clear our body of things like this. So that's the acute phase model. So we have this situation where we have a physiological system that is responsive to 2 domains of stimuli, one being pathogens and the other being body fat. And they share common physiological pathways related to cytokines, which lead to the production of the activation of innate immune pathways, as well as perhaps increasing risk for cardiovascular disease. Now the problem is we only study the system in people like this, right? So we're looking at this pathway, but the way the system functions, the way it has evolved, the way it functions in other mammals and vertebrates, involves primarily these kinds of pathways. So in studying this system only in relatively privileged, affluent, post-epidemiologic transition settings with high levels of overnutrition and low levels of infectious disease, we artificially constrain the range of environmental variation, which may be critical to understanding the system in a more comprehensive way. And so for that reason, I've been looking at inflammation and its correlates in Bolivia, the Philippines, and in Ecuador, and as well as the United States. And I'm going to talk about two studies that we've been doing, one in the Philippines and one in Ecuador. The Cebu Longitudinal Health and Nutrition Survey began in the early 80s, started by Barry Popkin, an economist at UNC Chapel Hill. Initially started as an investigation of maternal and child health and the predictors of child survival and breastfeeding and growth in low-income developing world settings like the Philippines. And it did that very well. It was a very important study in demonstrating the health benefits of breastfeeding at a time when formula use was on the rise. And then the study ended. It was designed to sort of end after that period. It began with the recruitment of more than 3,000 women from 30 randomly selected neighborhoods outside of around Cebu City, which was the second largest metropolitan area in the Philippines. Every woman in the randomly selected neighborhood who was pregnant during a fixed window of enrollment was asked to participate, and over 95% of them did sign up. And they were interviewed, measurements were taken of them and their homes, their neighborhoods, and then at the delivery of their babies were weighed, and then every 2 months for the first 2 years of their offspring's life, they were visited in the home, the baby was weighed, length was measured, information was collected on breastfeeding, diet, how the mom was doing, levels of infectious disease, things like that. So the study was designed to end there. But the study was done in a really nice, full collaborative partnership with colleagues at the Office of Population Studies at the University of San Carlos in Cebu, many of whom have PhDs from institutions in the United States. And they kept touch with the participants, did a couple of surveys in 91, 92, and 94, 95 to see how the kids were doing. There's some nice work on educational attainments of kids in relation to these kinds of exposures. And then here in 98 and 99, when the kids were 14 and 15, I was doing a postdoc at UNC Chapel Hill. And this is when I became first involved with the study. And this was also about the time that this idea that we really needed to pay more attention to early environments as predictors of health and physiological function in adulthood, this started to come online. And so we realized quickly that this data set was among the best, perhaps in the world, for looking at these kinds of questions, given the high resolution of prospectively collected data that we have in infancy. So we did a little bit of work here, but generated a bunch of projects to do a major follow-up at 2005, where we could find 1,885 of the original participants who were in their early 20s at the time of the survey. We did a full social, psychosocial, demographic survey of them, and we also collected blood and saliva samples. And these are some of the things that we measured in those samples, and I'll talk about a few of those in a moment. Okay. So this is what CRP looks like in the Philippines compared to NHANES data, commonly accepted as sort of the gold standard community-based study of health in the United States. And what I'm showing you here is data from NHANES, age and sex matched. So these are young adults in the United States. And overall, the level of CRP in Filipino young adults is 0.2 or 0.3 milligrams per liter compared to 0.9 milligrams per liter in the United States. And consistently in the U.S., we see a sex difference where women tend to have higher CRP than men. That's not the case in the Philippines. So I'm not sure what you're thinking when you look at this, but when I first saw these data, and many of my colleagues, I was talking to some colleagues about this seemed counterintuitive to us. Here we have the Philippines, which is an environment with higher burdens of infectious disease than the United States. It is in the midst of the global nutrition transition, so rates of overweight and obesity are on the rise. But infectious disease is still the #2 source of mortality in the Philippines. So there is a burden of infection that is higher than in the United States. So if CRP is involved in inflammation, maybe you might expect it to be higher. But that was clearly not the case. One obvious possibility, based on what I've said before about the role of body fat in promoting the production of CRP and activating inflammatory pathways, is differences in body fat distribution. Americans are heavier than Filipinos, who tend to be lean, although, as I mentioned, that's changing. So in 2005, the average waist circumference for participants in our study was 70 compared to almost 89 centimeters for young adults in the United States. So maybe that explains the population difference. Here we have that comparison based on waist circumference groups. So even if you stratify by waist circumference, at that same level of body fatness, Americans produce more CRP than folks in the Philippines do, at least the folks in our study. So there's something else going on here. And my interests are particularly focused on evaluating whether early environmental factors might explain this. But there is another possibility, and I always used to get this question. I'm very happy to have an answer, that genetic differences across the populations cannot explain the differences in CRP. So inflammation, there are SNPs and alleles that matter to individual differences in inflammation. That's been pretty well established. I have a colleague at UNC Chapel Hill, Karen Mulkey, who does genetics of complex diseases and has become a collaborator on this project. And in 2005, she used the DNA samples and ran panels looking for genes related to obesity, cardiometabolic diseases. And these are genes that have been previously associated in the United States and in Western Europe with higher or lower levels of CRP. And we find a very similar pattern of association in the Philippines. We explain small amounts of variance in CRP, 4 or 5%, and there are not dramatic population differences in the level of pro- and anti-inflammatory SNPs across the two settings. So the genes matter, but not to the story that I'm telling you here today. All right, so do early environments matter? So the model is that I'm going to take you through here is that we know as an adult your level of body fat, your level of pathogenic exposures, how likely you are to be exposed to an infectious agent. Both these things matter to your concentration of CRP, as well as some health behavior things, which I won't really talk about, but I control for. So might there be analogous exposures early in life, in infancy, that may program the system or have a direct effect in the system in some meaningful way? So this is the model that's guiding my selection of variables. And in terms of timing, when we're looking at these exposures, a priori, there are very good reasons to think that the first year in particular, but probably the first 2 years of life in infancy are critical periods of development of the immune system. And this figure shows you that. This is the point to take away. And what the top figure is an enumerative representation of where aspects of the immune system are throughout the life course, and this is a functional measure. So I'll talk you through it. On the x-axis is age, going from birth to 20 years of age. The y-axis is percent of adult level. So 100 here this is where you're at age 20. As an infant, your number of T cells and B cells are 2 to 3 to 4 times higher. The number of cells circulating in your body, despite the fact that you only weigh 10 pounds, the actual absolute numbers of cells are dramatically higher. The size of the thymus is double in infancy than what it is for us in this room right now. And the function of these cells, of the lymphocytes in particular, is upregulated. They're very sensitive to activation, and that's what this is showing you. If you prime these cells, they will produce cytokines. They will replicate and divide. So there are aspects of the immune system in particular, of the specific immune system, that are very active in infancy. And this makes sense if you think about what the immune system has to achieve. You're being born into an infectious disease ecology that you can never anticipate, because microorganisms evolve so quickly on the order of days or perhaps weeks, whereas humans evolve across generations that are 20, 15 to 30 years long. So we can't compete on that level. So we have to learn through a series of very cool processes that sort of embody Darwinian processes within our own bodies. We have to learn about our infectious disease ecology. And that's what's going on in infancy. That's why my interpretation of why these processes are really upregulated. So this points to infancy as a critical period of education of the immune system, where environments may have disproportionate long-term impact. So that's in part why I'm looking in infancy. Also, I have data for the first 2 years, so that's an important part of why I'm doing that. So this is what we have in terms of operationalizing and testing some of these hypotheses. From the study, we have measures of birth weights measured, ponderal index, length for weight, whether the gestational age and whether the baby was delivered preterm or not, parity, multiple measures of socioeconomic status for the mom, the partner, the household, and the community. And then in infancy, we have those measures as well, but we also have weight gain and length gain of the infant, duration and pattern of breastfeeding, multiple measures of household cleanliness and sanitation, which I'll get to in a minute, and self-reported or mom-reported symptoms of infectious disease in the infants. And then we have comparable measures in adulthood that prior research in the U.S. and other settings has shown matter to inflammation that we can control for. Some of these may be on the causal pathway, but the results I'm going to show you don't change whether we include these or not. So this is what we found with respect to birth weight and CRP in adulthood. individuals who were born with larger birth weights have lower CRP as young adults. And I'm modeling here the probability of elevated CRP. The results are the same if I present the actual CRP concentrations. If there's a question, I can tell you why I decided to model probability of elevated CRP. It has to do with the distribution. But the results are essentially the same. So as birth weight goes up, probability of high CRP goes down. And this is the first study to show this in a prospective birth cohort. And it's potentially important because there are lots of studies that have shown that birth weight is associated with cardiovascular risk in adulthood. So this is consistent with that and suggests that inflammation could be a mechanism through which that association is operating. But this cannot explain the population differences in CRP across the U.S. and the Philippines, right? Filipinos, in general, give birth to lower birth weight babies, particularly back in the early 80s. So if anything, they should have higher concentrations of CRP compared to Americans. So this is not part of the bigger story that I'm trying to tell you here, although I think it is important. Diarrhea morbidity in infancy is an important predictor of CRP. So this is, I think, more interesting. And what I'm showing you here is that this is the number of intervals. The interviewer went into the mom's home, the baby's home, and asked the mom, over the preceding 7 days, has your baby experienced symptoms of diarrheal infection? And this is the number of intervals in infancy during which the mom said, yes, this has happened. And so it's not a complete census of diarrheal morbidity in infancy, but it's going to be highly correlated with that. And individuals, babies who had higher levels of infectious diarrhea in infancy have lower CRP as an adult. Interviewers went into these homes and they observed the homes, and one of the things they looked for was whether babies were at a stage where they were crawling on the floor and whether animal feces was present on the home. Many of these homes were open. This is a tropical environment. Pigs, fowl, dogs were in some cases allowed to roam freely in and out of the home, so it was not unlikely that babies would come in contact with animal feces. So this is a proxy measure for the likelihood of that happening. The number of intervals, the number of times the interviewer came into the home and saw that this situation was happening. And a higher frequency of contact with fecal material, lower CRP as an adult. It's also a highly seasonal environment with a rainy season and a dry season. And that seasonality is also associated with seasonality and infectious disease burden. And a lot of the early work on this study was associated with seasonality and trends in infectious diarrhea and how breastfeeding can buffer infants from that situation. Kids who are born, infants born in the dry season, when infectious burdens are lower, actually experience more infectious diseases in their first year of life because they're alive for more of the non-dry season, right? It's a little counterintuitive, but kids born in the dry season, if you look at how that associates with infectious disease exposure, they have more infectious diseases. And they also have lower CRP as an adult. Okay? So we have 3 measures of microbial exposure in infancy. Each on its own I wouldn't put too much stock in, but they coalesce on a similar conclusion, and they each have different sort of levels and strengths to them as measurement constructs. And they suggest that lower levels of microbial exposure are associated with higher CRP, and that's independent of prenatal undernutrition or lower birth weight. And the mechanisms of this are not known. This is something that we're starting to look at, and I'll talk about a little bit in a moment. But one possibility is that there's higher level of pro-inflammatory signaling, IL-6 in particular. IL-10 is a very important anti-inflammatory cytokine that actually actively disrupts the production of IL-6. So it could be that you have different levels of anti-inflammatory signal. And there also could be changes in the phenotypes of T cells in particular, like T regulatory cells that produce a lot of anti-inflammatory action. So mechanistically, we don't know exactly what's going on. If there's any immunologists or molecular biologists or microbiologists in the audience, maybe you have some other ideas. Conceptually, which is where I like to fly, this is what I think is going on. So in a high pathogen environment, you can imagine these red arrows as microbial challenges, right, and exposure in infancy during this critical period of immune development. And so you get a challenge, the system becomes activated, and then it turns itself off. And you do that over and over and over again. So you have lots of opportunities, lots of educational opportunities for your system to develop in response to the local infectious disease ecology. And then to the extent that this is actually a critical or sensitive period phenomenon, those pathways carry forward into adulthood. And then when you're exposed to a pro-inflammatory stressor in adulthood, you mount a quick and efficient response that then becomes shut down when the infection has resolved or the issue is resolved. So it's a very efficient on and off kind of dynamic. If you're in a more sanitary, hygienic, low pathogen environment, you have far fewer opportunities for this kind of educational process to unfold. And then perhaps as an adult, lacking those well-educated regulatory pathways, when there is an inflammatory stimulus, the system activates, becomes overactivated, or is slower to turn itself off, or it just kind of meanders and is not very acutely regulated. It's sort of always on at a chronic level. So is there any evidence for this? So this is my interpretation of why there may be lower CRP in the Philippines, but do we have any evidence to support that? So one source of evidence is that in those 2005 blood samples that we measured the CRP in that I just told you about, we also measured IL-6 and IL-10 as pro- and anti-inflammatory cytokines, respectively. And the dots here show you previously published values for populations all over the world, but mostly in Western Europe and the United States. These are values from healthy adults, right? And there's a ton of work using clinical populations. So we a graduate student, spent a lot of time mining the literature and finding studies that had sort of healthy controls. And we summarized that. And the average IL-6 concentration across these studies is 1.9 milligrams per liter. And the average IL-10 is 3.17. In the Philippines, IL-6 is lower and IL-10 is higher. And this, I think, is particularly interesting. This is quite a bit higher. So there could be a different balance of pro to anti-inflammatory cytokine signaling in the Philippines that may explain the tight control over inflammation and the lower levels of C-reactive protein. So that's some preliminary support, at least my interpretation, for this model. Going forward, we wanted to do a study of CRP biovariability in an environment characterized by higher levels of infectious disease. And this is what we did this in Ecuador last summer. And I did not go to Ecuador, but one of my graduate students did. And this is the model in the United States, the chronic low-grade inflammation model that I was telling you about. So if you are born into a low pathogen, very sanitary environment, then we know, at least correlationally, as an adult, you're going to have a pattern of CRP production like this, with lots of between individual variation and stable, relatively stable within individual variation over time. We have no idea what those dynamics look like in a pre-epi-transition population. There are two studies that have looked at this in the United States, two studies of CRP biovariability, period, that I have found, both in the United States, both with the same conclusion. But what does it look like in the Bolivian Amazon or the Ecuadorian Amazon? We don't know. It could be that we get a pattern like this. It could be that we get a pattern like this. It could be that we get this pattern, but everything's shifted up. Which is in fact what the cohort morbidity hypothesis would suggest. So we wanted to figure that out. So we designed a study and implemented it in Ecuador. To orient you to the results I'm going to show you in a minute, this is from one of the studies of CRP biovariability in the United States. These are individuals on the x-axis and their CRP. And this is over a 6-month period. Each dot represents a CRP value for that individual. So each individual has multiple samples, and this line represents 3 milligrams per liter. That's the commonly used cut point to identify high-risk CRP concentrations as opposed to average or lower risk. And you see that there's separation here, right? There are individuals for whom CRP is consistently above this 3 milligram per liter threshold, and there are a number of individuals who flirt with that line down here. But there's a lot of, again, between individual variation and not as much within. So what do we see in lowland Ecuador? This study we did with the Shuar. They're an indigenous Amazonian population in the Amazon basin, so the Amazon side of Ecuador. Andes Mountains run here, and much of Ecuador, much of the research in Ecuador is done in the highlands, in the mountain region, but the actual Amazon basin of South America begins on the east coast of these mountains. And so this is the very westernmost edge of the Amazon basin, tropical rainforest. They live in a system of rivers, very few roads, no running water, no electricity, and high burdens of infectious disease. They're basically horticulturalists, a little bit of hunting and fishing. High degree of child death due to infectious diseases, and the main sources of morbidity, even for adults, are acute respiratory infection, gastrointestinal illness, and vector-borne disease. Sounds like a fun place to go. That's why I didn't go. Okay, so we did this study with 52 adults, 18 to 50 years of age, 4 weekly sampling intervals. So we visited them every week to the day. And we collected a dried blood spot sample. So this is related to one of the major areas of my research, which is the development of minimally invasive methods that allow us to study human physiology in places like the Bolivian Amazon or the Ecuadorian Amazon. We collect the sample by nicking a person's finger with a lancet, commonly used by diabetics, similar kind of process, except we put the blood onto this filter paper, which is designed for neonatal screening applications in the United States. The blood dries on the card and becomes preserved, even at tropical temperatures. And then we ship the samples back to the lab where we punch them out with a hole punch and put them in a buffer. We basically make whole blood again. And then we add them to an assay plate and we do a sandwich enzyme immunoassay just like we would do with serum or plasma. And then we generate a calibration curve and we generate very precisely the concentration of CRP in each of these samples. And this kind of data collection or sample collection approach allows us to gain access to people in places like the Ecuadorian Amazon and to go to them every week. And get a blood sample from them. So it facilitates the kind of research questions I'm interested in answering. And here's what we found. So again, we have participant on the x-axis, CRP concentration on the y. Here's the 3 milligram per liter threshold. And each diamond represents a value for that participant. So the participant over here on the right has values stacked up like this. And I've sorted the values based on sort of the overall average concentration across individuals. Okay. Does this look different to you? There are none of those, right? There are no individuals who consistently produce CRP above 3 milligrams per liter. In fact, only a third of the individuals, 17 people, produced one CRP value above this threshold. And on the other weekly intervals, they produced values that were very low, in most cases less than 1 milligram per liter. In fact, most people produced a value less than 0.5 milligrams per liter. So it's a very different pattern of variation. There is, in fact, no chronic low-grade inflammation in this setting. There's only acute activation. There's another way to represent this. This is going across time, so week 1, 2, 3, 4. And these are the 17 individuals for whom at least one sampling interval had an elevated CRP. So if you take week 2, you can see that there are about 6 individuals who have elevated CRP, but the next week, every single one of those individuals has a value that's less than that high-risk threshold. So if you were doing a cross-sectional study and you came into this setting and sampled at weekly 2, you would say, oh, about a third of this population has high-risk CRP. That matches up to the U.S., where about a third of the population also has high-risk CRP. But you would misclassify every single one of those individuals by virtue of doing a cross-sectional study design. So to my mind, that is actually pretty compelling evidence for a different set of regulatory dynamics, that individuals in the Ecuadorian Amazon can produce high CRP, but also very low CRP. And in the United States, it seems like people can produce high CRP, but a lot of people cannot produce that low value. And those are the individuals who are probably at elevated risk for cardiovascular disease. And I trace that back to exposures early in life, setting up the dynamics of the regulation of this system. So if this is true, we can talk about whether I've convinced you or not. This does have some implications for how we understand inflammation and its implications for other things we care about. And so maybe we shouldn't think about early environments mattering directly to inflammation in adulthood, but think about how early environments set up the dynamics of a system, how they set up how the system is regulated, and the consequences of differential phenotypes, differential patterns of regulation matter in response to stressors in adulthood. And in orange here we have a set of pro-inflammatory stimuli that are well established and in many cases of high interest to some of us. Body fat, I talked about, upregulates inflammation. Smoking is a well-established pro-inflammatory stressor. Vaccine is a very nice experimental paradigm for activating an inflammatory response, and I'll talk about that more in a minute if we have time. An infection activates inflammation. Pregnancy is an inflammatory event. I'll talk about that in a second. And there's a lot of interest in psychosocial stressors and their impact on inflammation. So perhaps all of these things are their impact on the inflammatory response and implications for disease may be moderated by how your body regulates inflammation in response to early environments. So I'm going to show you some preliminary data that suggests that's the case. Going back to this original observation that in the United States we produce more CRP than Filipinos do per unit of body fat. Well, it could be that in the Philippines, based on higher levels of exposure to microbes and things like that early in infancy, there's a more robust set of anti-inflammatory pathways in process. So even though body fat is a pro-inflammatory stressor or a pro-inflammatory input, your body is better able to keep that stimulation in check. All right? And perhaps that's why we have lower CRP there, even at the same level of body fat. And if we look at this with our data in Ecuador, we'll find the same thing. Smaller sample here, only 50 people total, so 10 to 15 people in each of these cells. But these are older adults, 18 to 50, and again, at every level of waist circumference compared to NHANES, we have lower levels of CRP in Ecuador than in the United States. Now, what about stress? So I've done some work on stress and inflammation and published a paper with John Cacioppo and colleagues using his data set of older adults in Chicago. And we found that perceived stress, Cohen perceived stress scale, positively associated and significantly associated with higher CRP among older adults controlling for everything. Okay, so that's interesting. And there are some good reasons to physiologically to understand how this may happen, which I can get into if there are questions. We also have the PSS. We administered it to our Filipino participants. And putting aside for the moment the question about the validity of that instrument in a Filipino cultural and linguistic context, I could still analyze the association between variation in perceptions of stress and CRP in the Philippines, and I've done that here. And there is a positive association, but it's very weak and it's not statistically significant. So that's interesting. But what if this other issue of early environments moderating the impact of stressors in adulthood, how might that inform what we do here? So I tried to test that explicitly, and I actually literally did this last week, so it's not fully fleshed out. But just took one of the variables that I showed before mattered to CRP, one of those microbial exposure variables that mattered to CRP in adulthood, and just ran an interaction. And so here we have perceived stress in interaction with season of birth predicting CRP. If you were born not in the dry season, so this means you had lower levels of infectious exposure in the first year of infancy, then you start to see a pattern of association that looks like Chicago. Higher stress, higher CRP. If you were born in the dry season, higher levels of microbial exposure in infancy, perhaps more robust anti-inflammatory networks in place, there's no association, and if anything, it's negative. Okay? So on their own, neither of these lines are statistically significant, but the interaction is. So there is a different pattern of association between these variables. So we could trace this back to early environments. Now, pregnancy. So pregnancy induces a lot of changes in a woman's immune system and how she regulates immune function. This is very important because the fetus is only 50% genetically related to her. So there has to be some strategic suppression of immune processes that would otherwise reject the fetus. And changes in inflammatory dynamics are part of this process. In 2005, when we collected our blood samples from our participants, there were about 80 women who were pregnant at the time of the survey. And we collected blood from them. But in all the analyses I've showed you, I've eliminated them from the sample because their CRPs are up and the dynamics are different. So we know that that's not part of what we're looking at. But that's actually a very interesting subset of women to look at. So I'm showing you very preliminary. These are pilot data that are going to feature in a grant application to justify additional research. So very preliminary. And what I've done here is taken women and divided them just on median splits on whether they had high or low CRP at the time of the blood collection and whether they had high or low IL-10. And I'm featuring IL-10 here because part of the story that I'm developing is that IL-10 is a very important anti-inflammatory signal. It's like driving down the highway at 100 miles an hour with your foot on the accelerator. That's the pro-inflammatory input. If your brakes are working effectively, then you can slow down and keep things in control effectively. And that's a better place to be, right? Better than going 100 miles an hour down the highway with no brakes. So no anti-inflammatory signal, no way to keep things in check. And I'm going to show you a slide about cardiovascular disease in a minute that helps me justify this approach. But here's the distribution among women who were born into a low microbe household. And I just took the variables I showed you before and did a summary thing and a median split, very coarse. About a quarter of the women have low CRP, so below median. About a third of the women have high CRP and low IL-10, so I'm interpreting this as an uncontrolled inflammatory state. And then about a third have high CRP and high IL-10. I'm interpreting this as a controlled inflammatory state. Now, if we look at women who were born into homes, this is when they were born, 20-some years ago. If they were born into a home with a higher level of microbial exposure, they're much more likely to be in this well-controlled inflammatory state. So does this matter? I mean, this is interesting. I mean, one of the things that's very cool about this to me is you're perturbing the system and you're showing that the dynamics of the response are really, it's very interesting. Pregnancy is the perturbation here. But does this have any health consequences? The thing that got me thinking in this direction is this study of cardiovascular disease. So these were patients who presented to a hospital with a heart attack. Their blood was drawn and they were followed for 6 months. And the outcome is death or subsequent heart attack. And at baseline, individuals who had low IL-10 and high CRP had the worst outcomes, far and away the worst outcomes. One in 5 of them was dead or had a subsequent heart attack. The people who had the best outcomes had high CRP. So this is consistent with the idea that inflammation is good for you in certain circumstances. It's part of healing. It's part of fighting infection. But they also had high IL-10. So the people who did best had inflammation, but a controlled inflammatory state. All right. So might this apply also to an important health outcome in middle adulthood for women, young adulthood for women, the birth weight of your baby? And this is what we find. So individuals in this well-controlled inflammatory state have an average birth weight that is more than 100 grams higher than women in the poorly controlled inflammatory state, or women with no active inflammatory activity. Again, suggesting that inflammation is doing something good here, but you want to control it in a certain way. So small sample size, a lot of caveats here, but enough, I think, to motivate us to pursue a more life course and even intergenerational approach to the study of inflammation. And this is our working model, and this is the life course here. And we have an inflammatory phenotype which develops in infancy, childhood, and into adulthood, and we have a lot of interest in how that matters to inflammation-related diseases of aging. But inflammation is critical in pregnancy, and pregnancy can be a key moment of transition of health across generations that sets up the next generation with respect to these processes as well. And we can trace an individual's inflammatory phenotype back to her infancy and prenatal nutritional environment, the postnatal infectious environment. And there's a lot of interesting research on the role of psychosocial stressors in childhood that I didn't talk about, mattering to the development of inflammation as well. And I have broader interests in social disparities and inequalities. These are all exposures that are partitioned in different ways by socioeconomic processes. I think I'll probably end there. I have 2 studies that we're putting in motion that basically test various aspects of this model. If there are questions on that, I'd be happy to feature them. But I want to leave time for questions, so maybe we should do that. Okay? Joe. Thank you. Almost all of the deliveries were in the home or a local birth center. Yeah, very few C-sections. But that, I think, is a very interesting possible route. I mean, there's a lot of, particularly with recent interest in microbiome and those kinds of, and transmission of microbes across generations, doing something like this in the United States, I think, would be very interesting to look at those kinds of birth circumstances as predictors of inflammation in adulthood. Yeah. Yes?
I meant to talk earlier today on inflammation.
Mm-hmm. Yeah. Mm-hmm. Yeah. Yeah. Mm-hmm. So I think there are two things there. One is there is a lot of research and interest in the United States in particular on infection and the regulation of inflammation during gestation, predicting primarily preterm delivery, but also lower birth weight. But in the United States, lower birth weight is almost always associated with preterm delivery. There's not a lot of intrauterine growth restriction. And that's a very interesting set of dynamics that I think matters here, but we just didn't have the data to address it. But with respect to level of prenatal and postnatal exposures that might matter to the story in the U.S. versus the Philippines, I think the Philippines in the 80s, the level of exposure to the kinds of microbes that I think matter here probably orders of magnitude greater than what happens in the United States, except maybe on a rural farm where you're really actively engaged with animals. I don't think this will necessarily track with social disadvantage, socioeconomic disadvantage, but that's a very testable hypothesis and something that I would like to look at. And a lot of literature on the hygiene hypothesis and the immune pathways related to that suggests that it's not necessarily overt infection. So of course, I have a 4.5-year-old, and every time he came home with a cold, I was like, great, that's one notch down on the risk for asthma. But it's probably not that. I mean, those things are correlated, but what's probably lacking is exposure to common commensal microorganisms that are in dirt and rotting vegetable material and water that's not been bleached. Things that have been part of the mammalian environment, let alone the human environment, for millennia, that in post-epidemiologic transition populations, and even not just post-epi, but post-epi hyperhygienic environments, that we've just totally eliminated from our diets, from our environments. So I think even in socially disadvantaged circumstances in the United States, people are mostly still drinking tap water, which they can assume is clean. They're not engaging with soil the way people in other parts of the world have or still do. So, but that is a testable hypothesis. So, Deb.
Sort of related to what you just said, what do vaccines do to all of this?
Yeah. Probably not much. So I don't think there's no evidence to suggest that vaccines really matter to the kinds of things I'm talking about. But if they do matter, they may actually put some individuals at higher risk for allergy and atopic disease and autoimmune disease. The way they activate the immune response is biased towards more Th2 kind of pathways, which have been associated with allergy and asthma. We do have information on vaccines in this study, but they're not as intensive and not as many. They don't seem to explain any variation here. So they do matter. They are not explaining what I'm showing here. But interestingly, along those lines of sort of what can we do to prime some of these pathways, there are some clinical trials in the UK where people as an adults who suffer from extreme forms of allergy or asthma or autoimmune disease have been given extracts of parasites or some of the microbes I'm talking about, and it's shown that it's alleviated some of their symptoms. And so I think that's interesting. My critique of that would be it's probably too late. If you really want bang for your buck, you have to do those things earlier in life when these systems are developing. So I'm impressed they show any action in adulthood, actually, but they do show some small effects.
Any questions? Any more questions?
Yeah.
Sort of related to some of the things that were said. I was sort of wondering if there's been any desire to try to find populations in the U.S. where you can do some studies of more rural or hippies or in some group where they were in the earth and more exposure to microbes.
This is something that I would love to do. I think another class of people to look at in this regard are immigrants, who may have grown up in a certain environment and come to the United States. If you can work through some of the selection issues there, that would be a very interesting case study. And there's a good opportunity here because there are a lot of studies, a lot of demographic, social science, population studies moving forward over the past 5 years, AdHealth, HRS, others, that are using these blood spot methods and are measuring CRP and have histories, retrospective histories of where people grew up, whether they went to daycare or not, whether they have older siblings, whether they were on a farm, immigrant. So we can actually test a bunch of these hypotheses. In fact, I'm eagerly waiting data from AdHealth so I can start to do some of those things. So, yeah.
So speaking of selection, is it, it's always
comparing these populations.
Yeah.
And the infant survival rate in the countries you're studying probably is way lower than in
Right.
the U.S.
Sure.
So
Sure.
how do you account for that?
Yeah, so there is definitely some selection. I don't worry so much about mortality selection in the Philippines study. There was some infant mortality, but it wasn't as high as I think the original investigators thought it might be. But I'm sure you noticed the original N was 3,300, and we were at 1,885 in 2005. And most of the loss happened in the first 2 years of life, and it was due to migration. It wasn't due to lack of participation or lack of enthusiasm for the study, but a lot of people moved out of Cebu. They moved to the United States, they moved to Hawaii, they moved to Taiwan, Hong Kong, and we didn't have the funds to follow them up. So the bias is in that we have people who are more likely to stay in Cebu, and so they're a little less educated and a little less mobile. Yeah.
I just wanted to follow up on the point about other populations, because there's actually some work going on primary prevention of asthma. You're probably aware of this, but people are looking at a lot of these kind of factors to try to tease out what you can do to prevent asthma, including looking at farm families. Most of that work has been done in European countries, but in the U.S. we're looking at Amish communities. So there are people who are trying to do some of those things, and a lot of interest in raw milk and other things with early exposure that can prime the immune system. Including gestational exposure to things like raw milk, which has its own risks.
Yeah, that's great. And I think we're, one of my goals is to get people to ask these questions and to generate hypotheses and test them using resources like that, which I actually was not aware of. There's a lot of research on the hygiene hypothesis in the context of allergy and immunology. I want to expand that out. If inflammation really matters to all these things that we care about, like cardiovascular disease and diabetes and dementia, then these same kinds of exposures early in life should matter. And so we should study them. Maybe it only applies in the Philippines and places like that, and not so much here. But we should figure that out, or try it at least. So. Yeah, Deb.
You were using kind of a cut point of 3. I forgot the units for CRP.
Mm-hmm.
Sort of above that is dangerous. Does that come from studies in the U.S. And if so, is that really valid for these other populations?
Right.
I know for things like BMI, the dangerous level differs among racial or ethnic groups.
Right. So yes and no. So I did use, it's based on data from the U.S., large epidemiologic cohorts, and it also sort of maps onto thirds of the population distribution. So that there's an official policy statement from CDC, the American Heart Association, that recommends the use of that cut point. But it's based on research in the United States. One of the reasons that I use this cut point approach in the Philippines is that the distribution has shifted so far left that I don't think 3 milligrams per liter is meaningful there. So I use tertiles. So whether you were in the high third of the distribution, and the cut point for that was 0.7 milligrams per liter. So that was just a way to think of relative risk. But I do think that the level really matters. And that is, in fact, one of the things that we've just been funded by NIA to look at among the moms. So the moms who were originally recruited into the study are now in their 50s and 60s and 70s, and they're aging into cardiovascular risk. So we will be able to, we've measured their CRP before, we're going to measure it again, and we'll be able to see what is the cut point that predicts risk in this environment. But we're also going to give them a vaccine. We're going to give them a flu vaccine, a subset of them, and measure the dynamics of the response to the vaccine and see if that's a better predictor of cardiovascular risk than just your average CRP level. So that's one of the things that I'm excited about launching. In fact, I'll be going to the Philippines in about 3 weeks to get that off the ground.
Thank you very much.
Thank you.